Hello students, my name is Niyati Seth and thanks for watching Edupedia word videos. My topic for the presentation is the 11th section of the chapter Internal Morphology of Flowering Plants. It is also known as Anatomy of Angiosperms. So in this section of the video, we will discuss about the anatomy of monocot stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheet. Okay, so let's see how it is. As I told you that for a better understanding of tissue organization of root, stem or leaves, it is convenient to study the transverse section of the mature zones of these organs. So here we are going to study about the monocot stem in the transverse section. Okay, we will observe it under microscope. So this is how it is. This is the transverse section of the monocot stem. Okay, see, the transverse section passing through the stem of monocot reveals the following details. Which are those? First is your epidermis, which is a single layer of parenchymatous cell, students. Okay, students, please note that epidermis is the outermost covering of the stem, which is represented by a single layer of compactly arranged barrel-shaped parenchyma cells. Intercellular spaces are absent. Can you see the intercellular spaces between these cells? No. So the intercellular spaces are completely absent in epidermis cells. Trichomes are absent too. A cuticle is present. Okay, a thin layer of cuticle is always present in monocot stem. Okay. The epidermis contains numerous minute openings which we call it as stomata. Okay, so as I told you that it is cuticle which is a protective covering on epidermis. Okay, now what it is? It is 3 to 4 layers of sclerenchymatous cells. Okay, hypodermis. Hypodermis is a region that lies immediately below the epidermis. Can you see that this hypodermis is lying just beneath epidermis? Okay, so this is the region that lies immediately below the epidermis, students. Okay, it is represented by few layers of compactly arranged sclerenchyma cells. Okay, they are compactly arranged, though some intercellular spaces are present. Okay, and they are made up of sclerenchymatous cells which are dead in nature. Okay, and it consists of 3 to 4 layers of sclerenchyma cells, students. Okay, so this is your hypodermis. Okay. Now, in monocot stem, it is seen that vascular bundles are conjoined, collateral and closed. I have already explained to you what is conjoined, collateral, closed. So, please refer to my previous presentation for it, okay, for better understanding. So, it is seen that in monocot stem, vascular bundle is conjoined, collateral and closed, students, okay. See, this is conjoined, okay. This is collateral, students. And what it is? See, this blue one is phloem. And this, the bigger round is metaxylem. And what it is? It is protoxylem, students. So, these three, they make up the vascular bundle. Okay, this is one conductive tissue and these two are one conductive tissue. So, these two are also known as vascular bundle. So, in case of monocot stem, vascular bundle is conjoined, collateral and closed. Okay. See, if cambium is not there, then it is considered as closed. If cambium is present between the conductive tissue, then that is known as opened. Okay. So, hence, no cambium is present, so it is closed type. Xylem is enlarged. Okay, because the metaxylem is present outside and the protoxylem is present inside. If protoxylem is present inside and metaxylem is present outside, then that condition is known as endark, students. Okay, what it is called as? It is endark. And if it is protoxylem is present outside and metaxylem is present inside, then that position is known as xylem exarch. Okay, what it is? The green one, these are the parenchyma cells around the vascular bundle, students. Okay, these are the connective bundles that connects the phloem, metaxylem and protoxylem. Okay, so this is the ground tissue. Ground tissue is a major component of the stem, students. It is undifferentiated. Okay, the ground tissue is represented by several layers of loosely arranged parenchyma cells, which encloses prominent intercellular spaces, students. The ground tissue is meant for storage of food. Okay. Now let's see what vascular bundle is. Vascular bundles, they are found irregularly scattered in the ground tissue. See, this green one are your ground tissue, and these metaxylem, protoxylem, phloem, they are your conducting tissue or vascular bundles. So they are found irregularly scattered in the ground tissue. Towards the periphery, the bundles are smaller in size, while towards the center, they are larger in size. Please note that towards the periphery, okay, the bundles, they are smaller in size. As you can see that this vascular bundle is present at the periphery. See, this is the periphery, okay, and vascular bundle which is present towards the periphery, they are smaller in size, while towards the center, see, it is almost near center, so it is larger in size, students. Okay, the smaller bundles are younger. See, vascular bundles, they are younger, while the larger ones are older. Okay, this vascular bundle which is present near the center, they are older, and that's why they are larger. Okay, hence the arrangement is described as centrifugal. That means the younger vascular bundle is towards the periphery and the older one is towards the center. Okay, so this kind of arrangement is known as centrifugal. Each vascular bundle has a covering called bundle sheet, which is formed by a single layer of sclerenchyma cells, students. Vascular bundle encloses both xylem and phloem. As I told you that it encloses xylem and phloem both. Xylem is found towards the inner surface. As you can see that this is the metaxylem and protoxylem which is formed inner to the surface area, and the phloem is towards the outer surface. See, this is phloem, this blue one are your phloem. So this phloem is towards the outer surface, as you can see. Okay, cambium is absent. Cambium is not present between phloem and xylem. That's why the vascular bundle is called as closed one. To be more precise, it is conjoined, collateral and closed. Okay. In the xylem, there are two metaxylem and two protoxylem vessels which are arranged in the shape of Y. Okay, as you can see that they are arranged in the shape of Y, students. Okay, two metaxylem and two protoxylem vessels, they are arranged in the shape of Y, students. Okay, this is protoxylem, this is protoxylem, this is metaxylem, this is metaxylem. So, two metaxylem and two protoxylem, they form a Y shape. Okay, the lower protoxylem, I'm talking about this one, protoxylem, is non-functional and it remains as a water-filled cavity called lysigenous cavity or protoxylem cavity. Okay, I'm talking about this one. Okay, that's why it is not represented by an oval shape just like this one, protoxylem. So that's a reason, because this is a water filled cavity, students. This protoxylem is water-filled cavity which is called as lysigenous cavity or protoxylem cavity, students. Okay. Xylem is described as endark because the metaxylem is towards outside and protoxylem is towards inside. That's why xylem is described as endark, students. In the phloem, only sieve tubes, I'm talking about this, okay? Only sieve tube, companion cells or phloem fibers are present. Phloem parenchyma is absent, students, in case of monocot stem. For example, maize, okay? So this was how it is. First is your epidermis, which is an outermost covering of the stem, which is represented by single layer of compactly arranged barrel-shaped cells. Okay, and they are parenchymatous in nature. Okay, that keeps on dividing, right? And there's no intercellular space present between the epidermis cells, students. Okay. Trichomes are absent. Can you see the trichomes? No. Trichomes are absent. Okay. And a cuticle is present. As you can see, this blue line is representing the cuticle, which is waxy substance. And it is also seen that epidermis of monocot stem contains numerous minute openings, which we call it as stomata. So there are the pores in the epidermis that we call it as stomata, students. Okay. Now come to the second layer. This is hypodermis. Hypodermis is represented by this red color. So it is a region that lies beneath the epidermis. See, this is the epidermis and this is the hypodermis. So it lies just immediately below the epidermis. It is represented by a few layers of compactly arranged sclerenchyma cells, students. Okay. Now come to the ground tissue. This green part, this entire green part is your ground tissue, students. It is the major component of a stem. It is undifferentiated. The ground tissue, it is represented by several layers of loosely arranged parenchyma cells. Can you see that these are loosely adhered cells? Yes. Okay. And they are parenchymatous cells. That means they keep on dividing. And ground tissue is meant for storage of food, students. Okay. Now come to the vascular bundle. This blue one is your phloem. This are your metaxylem and protoxylem. This is protoxylem. Okay. So vascular bundle, they are found irregularly scattered in the ground tissue. Towards the periphery, as I told you, that bundles, they are smaller in size, while towards the center, see this one, they are larger in size. The smaller bundles, they are younger. I'm talking about this one, and this is older. Okay, and this kind of arrangement is known as centrifugal. Each vascular bundle has a covering called bundle sheet, which is formed by a single layer of sclerenchyma cells. The vascular bundle, it encloses both xylem and phloem, students. Xylem is found towards the inner surface and the phloem is towards the outer surface, students. And cambium is not present. Okay, no cambium is there. That's why vascular bundle is called as conjoined collateral closed type. Okay, in the xylem, see, there are two metaxylem and two protoxylem which are arranged in Y shape, but the lower protoxylem, it is non-functional and it remains as a water-filled cavity, which we call it as lysigenous cavity. I'm talking about this cavity. Okay, it is called as lysigenous cavity, students. Okay. Xylem is described as endark because the metaxylem is present outside and protoxylem is present inside, students. Okay. And what about phloem? Phloem, only sieve tubes and companion cells and phloem fibers are present in case of monocot stem. But phloem parenchyma is absent in monocot stem, for example in maize. Okay. So this was the anatomy of monocot stem from out to in. Okay. Now let's see the transverse section of monocot stem. This is the epidermis. This is the hypodermis. These are the vascular bundles. These blue ones are your phloem. This is metaxylem and this is protoxylem, students. Okay. See, the smaller ones are the younger ones. The older ones are the larger ones, okay, and they are present towards the center, and this younger ones, they are present towards the periphery, students. That's the difference, okay. So this blue one is your phloem, this one is your metaxylem, and this is protoxylem. And I haven't shown the second protoxylem here because two metaxylem and two protoxylem, they are arranged in Y shapes, because the last protoxylem is water filled cavity, so I haven't shown here, okay. So don't get confused. So this is your phloem, this is your protoxylem, which is present towards the center, and this is metaxylem, okay. Please note that in monocot stem there are two protoxylem and two metaxylem which are arranged in Y shape, but I haven't shown the second protoxylem here, so don't get confused, okay. And what it is ground tissue, okay. Ground tissue is the major component, as I told you, and it is undifferentiated too. The ground tissue is represented by several layers of loosely arranged parenchyma cells, and thus it encloses prominent intercellular spaces, students. The ground tissue, it is meant for storage of food, okay. So that's how it is, the transverse section of monocot stem from out to in. First is your epidermis, which is the outer layer. Second, this red layer is your hypodermis, which is made up of sclerenchyma cells, and that lies just below the epidermis, okay. And they are compactly arranged. There's no intercellular space present between the cells. And then comes your vascular bundles, as I told you the entire process, how it is and how they are arranged in the centrifugal manner. And this is the ground tissue, which forms the major component of the stem, and they are undifferentiated, okay, and they are loosely arranged parenchyma cells. The ground tissue is meant for storage of food, students. See, this is the ground tissue, okay. So vascular bundles are numerous and they are scattered in the ground tissue, okay. Anatomical difference between the two types of stem. So this is the dicot stem and this is the monocot stem, students, okay. So this is the transverse section of the dicot stem and this is the transverse section of monocot stem, okay. See, trichomes are present. Can you see the long unicellular hair present on the surface of epidermis? Yes. So trichomes, they are present in the case of dicot stem, but trichome is absent in case of monocot stem, students, okay. See, hypodermis is made up of collenchyma in case of dicot stems, but in case of monocot stem, hypodermis is made up of sclerenchyma cells, ground tissue, cortex, endodermis, pericycle, medullary ray, pith, okay. These are the features of dicot stem, but in case of monocot stem, ground tissue such as ground parenchyma, okay, and medullary ray is present in case of dicot stem, but in case of monocot stem it is not present, okay. In dicot stem, vascular bundles are of which type? Conjoined collateral open type, okay. Conjoined collateral open type, students, okay. See, this is how it is in conjoined collateral open. And in case of monocot stem, vascular bundles are of conjoined collateral closed type, okay. See, they are considered closed because there is no cambium present. When the cambium is present between the two conductive tissues such as your vascular bundle such as phloem and xylem, then that is considered as conjoined collateral open type of vascular bundle. But here in case of monocot stem, vascular bundles has no cambium in between. So that's the reason it is called as conjoined collateral closed type, students. Okay. And before that, I will tell you the diagnostic feature of the monocot stem. Like, how can you identify it, students? See, there is no trichome present in the monocot stem, students. Okay. Presence of stomata. Okay. Stomata is present on the epidermis. Okay. And the third diagnostic feature is presence of hypodermis made up of sclerenchyma and presence of undifferentiated ground tissue, the green one. Okay. The presence of the numerous vascular bundle which are irregularly scattered with centrifugal arrangement, students. Vascular bundles, they are conjoined, collateral and closed with the endoxylem. Presence of only 2 protoxylem and 2 metaxylem vessels in each bundle. Presence of a lesigenous cavity is the characteristic feature of monocot stem. And absence of phloem parenchyma and presence of bundle sheet which is made up of sclerenchyma. So these are the diagnostic features of monocot stem. So this was all about the monocot stem. In my next section of the presentation, we will discuss about the dicot stem. So till then, stay tuned and keep watching Edupedia word videos. Thank you.